joined by Kelsey McCune, who is coming down from Santa Barbara. She is currently a postdoc there. She's a behavioral ecologist, and her postdoc is in the Institute for Social, Behavioral, and Economic Research, working with Karina Logan. Today, Kelsey is going to be talking to us about her research with the great-tailed grackles, and her talk is entitled Movement, Exploratory Behavior, and Range Expansion in Great-tailed Grackles. So please do welcome her here to Beth.
Thank you guys. Thank you for inviting me to come here today. I'm pretty excited to talk to you about this work. It's very early on. This is the first time I've presented it, so I'm excited to hear some feedback and impressions. So I am a postdoctoral researcher at UCSB, and I work in collaboration with Dr. Karina Logan, who is at the Max Planck Institute for Evolutionary Anthropology. They have been the ones to very generously fund all of this work. So the overall Grackle project is this like massive cross-population study of a bunch of different cognitive and behavioral traits in the great-tailed grackle that might help to explain the rapid range expansion. Today I just want to focus on one piece that I created for the Grackle project. It's my favorite part. So today we're going to talk about movement behavior, exploratory tendency in relation to the range expansion in this species. I was talking with Brian before we came into this room and mentioned the workflow for the Grackle project. It's a little unique, and he thought it might be interesting to some folks. So I want to go ahead and talk about that just briefly. On the Grackle project, we try to have an open and verifiable workflow. So what that means is that we try to avoid some of the problems that we're seeing in science that leads to the work being not reproducible. So what we do is for each component of our research, we pre-register our ideas. So we pre-register the hypotheses, the predictions, our methods that we're going to use, and even our analysis plan, including specific models. This then gets peer reviewed. So we get feedback on those things, and we make changes if appropriate. And we do all this before analyzing any of the data. Sometimes we are still in the process of collecting it. You just state where you're at in the research so that they know your level of bias. And so after it's approved and you've collected all your data, then you analyze it with the methods that you set in your pre-registration. And for the Grackle project, I'm not sure how consistent this is across different people using this, but for the Grackle project, all of our information is on GitHub, and anybody can look at it. And so the methods and all of our details for our pre-registrations are in GitHub and version tracked. So everybody can see any changes that we've made to our ideas, to our methods, and when those occur. After you analyze the data, you do the post-study write-up, and you submit it back to the place that hosted your pre-registration. And then they take a look at it, and they basically look at whether you did what you said you were going to do, that your methods and analysis plan matched up with your pre-registration, and theoretically, essentially, they kind of give you a thumbs up, and you're free to go and submit this work to be published in another journal. They often have agreements with specific journals where there's not an additional post-study review. Or you can publish it in the pre-registration website's journal. We use PCI Ecology right now, Peer Community in Ecology. So that's a very brief overview. I have a lot of thoughts on it, and as I was telling Brian, as an early career researcher, I'm happy to answer any questions, share any experience if you guys are interested in doing it. But otherwise, we can go ahead and jump right in. So as I was saying, the Grackle project is actually a very large study that involves many things other than what you see here. And in fact, some of these, let's see if I can get the slide to progress. Some of these have been done in collaboration with researchers right here at UCLA, so I wanted to kind of plug the work that they have done. We looked at causal understanding in the grackles as well as their inhibition. And we taught them to use touchscreens, so we also wrote about that as well. So just as a heads up, there are peers right here on campus that you could talk to about Grackles and their cognitive abilities if you're interested. Okay, so back to the topic at hand. So as we all know, humans are rapidly changing all aspects of the earth around us. So while to us it might seem like it's taken a really long time for humans to change an environment that looks like this into, for example, what we now know as New York City, on an evolutionary timescale, this is happening too quickly for most species to evolve adaptations to be able to persist in this kind of novel human-modified landscape. Consequently, one of the main outcomes is a decrease in native fauna in these landscapes and an increase in non-native fauna. Non-native fauna are able to come in because some of the ecological barriers that previously existed when it looked like this have been broken down. So they're able to become established when otherwise they weren't. But not all native species become locally extinct, and not all non-native species are able to become successfully established. So to understand who is able to actually persist when we change the environment, we need to understand what are the differences in these species that might permit this to happen. And so because these changes are happening too rapidly for evolved adaptations to be the answer, it's possible instead that differences in behavior among individuals and species might be what allows some to persist in human-modified environments where others fail. So one way to understand which traits might be facilitating persistence in landscapes that have been rapidly changed by humans is to look at species invasions. So species invasions actually occur over many different stages. So usually you have a transport stage where a non-native species is carried outside of its original range, either intentionally or unintentionally, to novel environments. They can survive and reproduce, and so become established in the introduction area. Sometimes they reproduce and they're able to make use of these new resources so well that they're able to spread outside of the introduction area into even more novel habitats. So for example, here in LA, maybe you guys know, there is the non-native yellow chevron parakeet. So this was introduced a while ago to supplement the demand for parrots in the pet trade. So they brought these parakeets over, they released them, they were able to survive in the wild. They did reproduce and make use of some of the resources here in LA, but they have never really been able to push beyond the boundaries of this introduction zone and to continue to invade into new areas. So in contrast, the American bullfrog is native to the eastern US. It's been brought out here to the west to California, introduced, survived very well, reproducing very well, and it has actually been able to spread beyond the introduction zone, and it's negatively affecting a lot of native amphibians. So in this example, this invasive species is doing much better at persisting and continuing to adapt to these novel environments as a result of human-induced rapid environmental change. So we can compare species like this to understand which behavioral traits might matter for persisting in human-modified environments. So there's a theoretical cluster of behaviors that is thought to help individuals and species progress through these different stages of the invasion process. And when you have two or more of these characteristics as an individual or as a species, it's called an invasion syndrome. And today I want to specifically focus on just two of these, mobility, which I will refer to as movement behaviors, and exploratory tendency. And I'm specifically interested in these two because they have been relatively less studied in avian range expansions. And it seems intuitive that when you are a species and you're trying to persist in a novel environment, the ability and the motivation to encounter conspecifics novel food resources, novel nesting habitat, or den sites would help to facilitate your survival and reproductive success there. Okay, so I already talked to you about how you can compare two different successfully invading species to look at certain traits that might have made one a little bit more able to persist than another. But today we're going to talk about within species variation. So to understand the traits that allow species to invade into novel habitats, when you look at this within species at different parts along the range, you can rule out other ecological answers like, are they omnivorous or are they frugivores, and is the new habitat suitable to that? Okay, so I want to break down a little bit more in depth what I mean by mobility in terms of the behavioral trait of movement behavior. So mobility can be broken down into different measurable ways that individuals move about in space. And the first and most popular that you guys might be familiar with, I'm not sure how many people know about these aspects of the movement ecology field, is home range size. So here we have an example of two individuals, one in green, one in yellow. And each of these individuals' sightings are indicated here with this dot, with dots. So you recite your specific individuals many, many times. And then once you have a good idea of the places that they like to hang out most, then you can project your home range area across those recitings. And usually home range only encompasses about 95% of the most common places where they are seen. So you're excluding some of these outliers where an individual maybe went once and it's a rare place to see them. So now you have this home range area and you can compare the size between and among individuals. So if your green individual really only likes to hang out in like a 1 block radius, then the size of the home range, the number that they have for home range, is going to be smaller than a yellow individual that might like to hang out across like a mini block radius. Okay, so another way to compare movement behavior among individuals is to look at the daily distance that they move. So you go out and you find your focal individual and you note their location at the start of your follow. You then follow the different places that they use in the amount of time that you set out to follow them, and you note the spatial location at the end of the follow, and then you measure the distance between these 2 points. And so you can compare this distance among individuals when the follow is the same length, and theoretically some individuals are going to travel a lot further in that time than others. And lastly, you might be able to compare movement behavior among individuals using turning angle distribution. So this kind of qualifies how individuals are moving about in space. So you have one individual here in green who's taken basically a straight line path. And if you look at the angle among 3 points, you can see that it's pretty obtuse. And this, when you have many of these points strung together and all of the angles are obtuse, you can say, okay, this individual pretty much just going straight line distance. So in contrast, you might have something like the purple individual where they're taking a much more convoluted path that day. So in movement ecology, they say it has high tortuosity. It's not a direct path. The angles in this path are much more acute. So the turning angle distribution here would indicate that this individual is doing a lot of route reversals. It's spending more time in a small area. Although, so home range size is one of the most popular ways to compare movement behavior among individuals, but we're not going to talk about that today until the very end of the talk. But I do want you to remember these other 2 movement measures that I kind of briefly overviewed. So movement behavior has been shown to relate to invasion success. So here we're looking at an example from the invasion of the cane toad into Australia. So this was a really well-documented invasion where they recorded the times that toads arrived in each area. So you can see the extent of the range here with the oldest portion. I guess I should use my pointer. The oldest portion of the range in the dark color, and then moving towards the newest portion of the range, or the border of the range, in a lighter gray. And so researchers in Australia tracked toads when they first arrived to their study site and looked at the movement behavior that these toads were exhibiting and compared it to toads that were present at their study site a couple years later. And they found that these toads were moving significantly differently. So the toads that first arrived, they called them the pioneers, were moving in these straight line paths, so they had very obtuse turning angles. They also moved very fast and far distances on their nightly tracks. In comparison, the toads that were present at the site a couple years later showed these more tortuous type of nightly paths where there were lots of acute turning angles. They didn't go very far distances. So there's a significant difference in the movement behavior based on the location in the invasion front for these cane toads. Additionally, movement behavior could be part of an inherent behavioral trait. And so in that case, the behavior, it varies among individuals that are at the same site at the same time. So here we're looking at an example from African elephants, where they found that the mean daily distance moved here on the x-axis was significantly different for each individual that they tracked. So individual elephant is on the y-axis, and it's sorted by their average daily distance moved. So they also found that, so what you're seeing here also on the plot is the distribution of the daily distances that these elephants are moving. And one other important thing here to indicate that this behavior is part of an inherent trait is that these, here it is. These daily distances moved are all clustering within an individual, and they're different among individuals. So this individual really tends to move in this amount of distance, whereas others tend to move much further distances consistently. They also found that males tended to move shorter distances than females, which is kind of interesting too. Okay. So lastly, it's possible that movement behavior is a result of is an emergent result of the personality trait exploration. So evidence for this comes from a study on human migration, where researchers looked at populations of humans from all over the world, and they were comparing the proportion of individuals that had a genetic footprint for a dopamine receptor type in the brain, and this dopamine receptor is related to highly exploratory behavior. So they were comparing these different populations all over the world, and they found that individuals that were historically from populations that had moved the furthest, so from Asia into North America and then down into South America, those individuals that made the longest macro migrations were also the most likely to have this genetic footprint for this highly exploratory phenotype. Okay, so one of my main goals for my research on the Grackle project was to combine these ideas to help to understand how this species of bird has been so successful in invading into new habitats across the US. So first I wanted to test whether movement behavior in the Grackles represented an inherent trait, and in birds they are much more able to fly greater distances, move longer distances more frequently. So I was curious whether it would also be an inherent trait in Grackles like it is in the elephants. Oopsies. I cannot see where my... Okay. Don't be too aggressive with moving the cursor, I think. I want to point out that when we talk about whether something is an inherent trait, what we mean by that is how repeatable is the behavior over time and across context. So we quantitatively decide if it's part of an an inherent trait by looking at the repeatability. And this means that there's low within individual variance in the behavior and high between individual variance over time. And so that's exactly what we're looking at here with this plot from the elephant example, where the behaviors were clustered, the mean daily distance move values were clustered within individual and different across individual. So there's low variance here within individual in the mean daily distance moved, and high variance between individuals. Secondly, I wanted to know whether movement behavior reflected the personality trait exploration. Similar to the research on the human migrations that I mentioned, are the individuals that are moving the furthest and moving the most also those that are most likely to show a high exploratory tendency? And lastly, I wanted to know whether there was variation in either of these behavioral traits in relation to the proximity to the range edge. So like we saw with the cane toads, are the grackles that are on the edge of the range, the pioneers, more likely to move further distances or in straight lines, or be highly exploratory compared to the grackles in the center of the range? Okay, so a little background information on the great-tailed grackle. This is the current range of the grackle, and for those of you who are not familiar, this is an eBird map. So eBird is a citizen science birding website. People submit observations of birds they've seen all across the world, and the data goes back pretty far in time, maybe the 70s. I'm not exactly sure. Anyway, so the grackles are the darkness of the color purple indicates the likelihood that you'll see a grackle in each of these given areas. So originally, grackles were constrained to these tropical areas down here in Panama, but they were able to push north over time through mountains and deserts in Mexico, the American Southwest. So they're pretty common here in Southern California. Although walking around campus, I didn't hear any, which I was kind of surprised by. So you guys will have to tell me where they are here. I know they're in Santa Barbara, but and they're even rarely seen in Canada. What makes this species ideal for this question is that their range is still expanding. So there is still a northern front, northern invasion front. So these individuals up here are still pioneers. They're still pushing into new habitats and experiencing new conditions. The range expansion was extremely rapid. So between 1880 to the year 2000, grackles expanded their range over 5,000%. And this was likely vastly facilitated by human-induced rapid environmental change, land use change. Grackles, I guess for those of you who don't know, this is a female grackle in brown. They're sexually dimorphic. And the males are in black, and they're larger in size. So they're opportunistic omnivores. They really excel at eating human trash. So that helps, but beyond that, they also have taken advantage of land use change like irrigation, agriculture, and management of livestock to be able to persist in areas that would otherwise be inhospitable, like the deserts in the American Southwest. So to understand how behavioral traits might have facilitated the range expansion in this species, we compare behavior in species at multiple places along the extent of the current range. We started our study in Arizona between 2017 and 2020. And so this is still a non-native population of grackles in Arizona. They arrived into Arizona in approximately the 1900s. So they've been there for a long time, had the potential to become locally adapted to those environments. So we call this the middle of the current range. We then moved our research site to Northern California, specifically outside of Sacramento. And on the West Coast, this is the furthest north that you can go to have a year-round large enough sample of grackles to be able to, answer some questions. And the Grackles actually only showed up in this area about 15 years ago. So we're constantly encountering people when we're walking around following the Grackles that are like, where did they come from? What are these birds? It's really strange. The original plan was to then move our study site down to the original center of the range in Panama, where Grackles, theoretically, where Grackles evolved as far as we know. But as everybody has experienced, the COVID pandemic pretty much put that on hold, possibly indefinitely. So stay tuned, I guess. So Grackles are not federally listed as an invasive species because they were able to push into new habitat on their own. There was no transport stage of the invasion process, but they do fall within the establishment and spread phase of this process. There is kind of a low human perception of Grackles in many places. They are agricultural pests, but it's really unknown the impact that they're having on native flora and fauna. So for all components of our research project, we need individually identifiable Grackles. So we catch them with a massive variety of trapping methods because they're very sneaky and suspicious. So we've used a bow net trap. This is a mist net. And we also have just a plain walk-in trap. As you can see, the bait that we use to lure them in is usually just Cheez-Its or goldfish crackers, something that's very colorful that they can see and high in fat. And then once we catch them, we put these different colors of color bracelets on their legs so that we can tell them apart. We take biological samples and some measurements. Most Grackles we then hold in captivity temporarily. We hold them individually in large aviaries or cages at our study site. And this is where we collect basically all of our data on the behavioral and cognitive traits. The only one that's relevant for the talk today is the measure that we used for exploratory tendency. So we use a novel environment assay to quantify exploratory tendency in each Grackle. This occurs after at least a week in the aviaries. So their aviary is a familiar environment. So then we introduce this novel environment, which is actually a small cat tent. So if you have a cat and you want to respect the wildlife outdoors, you can take your cat outdoors by putting it in this tent. And we look at how willing the Grackles are to approach the tent, how much time they spend in vicinity of the tent. Importantly, we do this in a way such that they have access to food and water as far away from the tent as we can get it. And this is so that the Grackles don't have to approach the tent. We only want to know if they are willing to approach the tent to investigate this novel thing. So we also always precede the novel environment assay with a familiar environment assay. So it looks just like this, except we take the tent out, and then we let the trial go for the same amount of time to collect a baseline of their activity within their aviary. And so this is a control condition for our novel environment. We want to, just one last thing, I think. We want to make sure that this method that we're using to measure exploration, an inherent personality trait, is actually measuring an inherent trait. So what I mean by that is we want to make sure that the performance of the Grackles with this task is consistent over time. So we repeat this assay after at least 2 weeks in a second trial. Okay, so this is a video clip from one of the Grackles from our middle population in Arizona. He comes down, he goes right into the tent. You can see his feet on the white floor of the tent to kind of locate where he is. These are 45-minute trials, and he spent at least 15 minutes just messing around with everything in and on the tent. At one point, he pulled a branch off of his plant in the background and was just also dragging the plant all over the tent. So I like to show this example as what it looks like in the most exploratory case. We had 19, I think, grackles that we measured for exploratory tendency in Arizona, and 4 of them went into the tent. This is from our Edge site. This is another male grackle interacting with the novel environment assay. He hesitated at the opening here of the tent for about 3 minutes. You can see his food and water at the front. The orange stakes are in the ground to measure the distance of his approach to the tent. So he went briefly into the tent, got immediately scared, and jumped back out. We did the novel environment assay on 24 grackles in our Edge population. Little foreshadowing. He was the only grackle that went into the tent, and that was the only time that he went in. So he spent very little time inside. So once they've completed our test battery in the aviaries, we want to release them back into the wild. But we want to be able to find them again to get our measures of movement behavior. So we give them a little backpack that has this special tag on it, which emits a high frequency radio signal. Each grackle gets a different frequency, so we can use this receiver and antenna to find them in the wild, to track them down, and then collect our measures of movement behavior. So I want to be able to tell you that we followed these grackles. You can see her color bands and her antenna with her radio tag, that we followed them through some really pristine natural areas and got to enjoy the peace and solitude of nature. But because they're grackles, it always looked a bit more like this, where we follow them around as they run under the cars in the Walmart parking lot, trying to eat trash, finishing the cheese dip from your Chick-fil-A order, scrounging in the bottom of your Walmart shopping cart for any goodies left behind, or just jumping straight into the dumpster to pull out any food that's mixed in amongst the trash. So we find them with the radio telemetry, and then we follow them around for about an hour and a half. And at 1-minute intervals, we use this iPhone app to drop a pin at the grackle's location. So here we go. So these red pins represent GPS points, so they have a latitude and longitude associated with them. And then the blue line in this figure is actually the experimenter's movement. So we always try to stay far enough away from the grackle that we're not affecting their behavior. But you can see on this day, this female was first foraging in the grass outside of the movie theater. Then she made me run all the way over between these 2 restaurants, and I found her again at the dumpster behind the Firehouse Crawfish. She spent some time exploring the trees in the parking lots, trying to steal food from other grackles, and ended up back over here at another dumpster at the AT&T store. So we do this with all of our grackles, and we repeat it across days, and we really try to do it for as long as their tags hold out. And so we end up with a ton of points like this. This is from our middle population in Arizona. We had over 15,000 points on the grackles there. Each of the dots is a GPS location of a grackle. The colors represent individual birds. So what I want you to look at here is the distribution of the colors on the map. So individual grackles tended to hang out in specific areas on the campus of ASU, Arizona State University, which was our study site, or in the surrounding neighborhoods. So you can see that they have these kind of distinct home ranges. We did catch all of these grackles on campus, so a few of them moved outside of our trapping area. They roost at night all together in like a communal roost. And the roosting sites were also on campus and in the neighborhood, so nothing too far away. They're not going too big a distances. Okay, So the scale of our operation at our edge population in California and southern Sacramento is pretty different. So it's occurring over a much larger area. We trap all of the grackles at one site just south of a large shopping center. At night, all of the grackles go to one roost site way down here. It's a marshy area, so they roost in the cattails. During the day, most of them do hang out at this large shopping center. However, some of them do go 5 miles away to this golden corral. But they will also go back and forth. So it's not just specific ones are spending all of their time here. You will see them again show up in the shopping center. The other thing I want to point out here is we have over 17,000 points in this figure, but you can't really see them because the space that the grackles are using in this population is completely overlapping. So they're not separating out distinct areas of the shopping center. All of the points for each individual grackle are pretty much piled on top of each other here in the shopping center. Okay, so as I mentioned, we follow them. We take GPS points at 1-minute intervals. And from these data, I'm going to explain, using this figure, the variables that we extract to measure their movement behavior. So each point has a latitude and longitude. Sequential points in time, so points that are 1 minute apart, you can think of as having a line connecting them. And this line has a measurable distance in space, and that's the step length. Among 3 points, like we talked about earlier in the talk, is a turning angle. So then we also extract the turning angles from each daily movement track, each follow that we do on the grackle. And this is the deviation from the straight line distance. So to look at the repeatability of these values within individuals, we compare their movement tracks collected across days. And so this brings us to the first question that I wanted to ask. Is the movement behavior in this species an inherent trait? Again, I just want to reiterate that what I mean by that is it repeatable across time? Is there low within individual variance and high between individual variance in this behavior across time? This is what variation in individual movement behavior can look like. So these movies animate a daily movement track for 2 different grackles. They are actually both playing at the same time. It's just that the individual in the movie on the left is a pretty sedentary dude. He's not moving very far in the same time frame that the individual in the movie on the right is traveling pretty quickly over this couple block radius. So this is a program that just animates the GPS points that we drop using that app. So let's talk about step length first. So again, this is the distance between points taken at 1-minute intervals. So I found for both the middle population and the edge population, average step length was significantly repeatable. I was also interested in how predictably these individuals are moving. So I looked at the variance in daily step length. So for example, if your individual is taking the same distance of steps within a day, then the variance is going to be very low. And if they're consistent in their movement behavior, then across days, they're going to continue to take always short steps. So when you look at the repeatability of the variance in step length, what we're seeing is that grackles in general in both populations are consistent in the step lengths that they take across days. They're consistent in the distance as well. These R values are repeatability values, and so that's a ratio. So they will always range between 0 and 1. To give you a reference, the repeatability value from the elephant example that I talked about earlier was 0.22. So the middle population grackles are showing like a little bit more individual consistency in their movement behavior, and the edge population grackles are showing a little less consistency. This plot is just illustrating those results. So we have step length distance on the x-axis, and then individual bird on the y-axis, sorted by average step length in the black dot. The bar is the interquartile range of observed step lengths. And so what you can see here is that there is a lot more variance, there's a lot more variation in the average step lengths in the Arizona population compared to the California population. And so this is how we have a higher repeatability value. But the repeatability value in California was high enough, there was enough clustering of step length values within individual such that we have a significant repeatability. Okay, so we're going to turn to talking about turning angle, with the pun intended there. So I first visualized the distribution of the turning angle data, and I found that all individuals at both sites are showing the same pattern of turning behavior. So here we're thinking about turning angle in degrees in a circle. So I hope you guys all remember your 8th grade geometry. And each individual's frequency of turning angles is here in a color, and then there's some degree of transparency so that you can see the others underneath it, and you would see any individual variation if it existed. And the x-axis ranges from 0 degrees, which means the Grackle continued in a straight line path, there was no turn, up to 180 degrees, which was a complete route reversal, so they went back in the other direction. And so all Grackles from both populations are tending to turn at around 150 degrees. So because the pattern is the same, there's not variation among individuals, turning angle is not repeatable. It doesn't seem to be part of a consistent inherent trait. Okay, so we found that the distance that individuals move and the predictability of that movement is repeatable within individuals over time, and represents, therefore, an inherent trait. But is that trait the personality trait exploration? So we'll move on to the second portion of the talk here. So when I looked at performance on the novel environment assay in trial 1 and trial 2, we found that performance for each individual was repeatable. So it seems that this method that we're using to measure exploration is measuring an inherent trait. I then used only the performance on the first trial and compared it to the step length values from the Grackle's movement behavior. But I found that there's no relationship between step length and exploration, either for the average step length or the variance or predictability in step length. For our middle population in black, or for our edge population in red, there's no relationship between these two traits, at least with the method that we're using to measure exploration. So some of you might already know what you're about to see here, but because there is no variance between individuals in turning angle, we wouldn't really expect any relationship with exploration, but I wanted to create this neat plot to show you another way to visualize turning angles. So what you're looking at here is the frequency of the turning angle is represented by the size of the wedge. High exploratory individuals spent more than 1 minute within 20 centimeters of the novel environment, and they're gray with no border. Low exploratory individuals spent less than 1 minute near the tent, and they're white with a black border. But these wedges all look gray with a black border because the values are completely overlapping for turning angle in both of these exploration types. Okay, so movement behavior is an inherent trait. That trait is not exploration, but it's possible that these two behaviors could separately have influenced the invasion success of the great-tailed grackle. So now we're going to compare the results that we got for these behaviors across our sites. So for step length, again, we have no difference in the average step length that individuals are taking or the predictability of the step length in either population. So Grackles in Arizona and Grackles in California are taking steps in about the same way, moving the same distances. As you already know from the previous plots that we've looked at, there's also no difference in turning angle for the middle or the edge population. So movement behavior may not have been important in the range expansion for the great-tailed grackle, but what about exploration? So I looked at the duration that individuals spent within 20 centimeters of the novel environment, compared that across sites. I also compared their performance on the familiar environment assay, and I found that there's no difference in baseline activity, which is good. When there's no novelty involved, these two sets of grackles are moving about their aviary in the same way. When we introduced the novel environment, I found that the California grackles, the edge population, are spending significantly less time in close proximity to the tent compared to the grackles in Arizona in the middle of the range. So seems like the California grackles are less exploratory. So just a bit of a rehash, we found movement behavior, at least in distance that individuals are moving, is consistent within individuals, like we saw in the elephant example. There was no relationship between movement behavior and exploratory tendency, at least as we measure it with the novel environment assay. Movement behavior seems like it did not facilitate the range expansion in the great-tailed grackles because the site in the center of the range and the site on the edge of the range are showing the same pattern. But we did find a role for exploratory tendency, or specifically a lack of exploratory tendency, in possibly facilitating the range expansion because the ones on the edge are less exploratory than the ones in the center. This is a little counterintuitive based on the examples that we talked about in the beginning of the talk, but exploratory tendency is not always a good thing. And in particular, there are some good examples from when animals have been translocated. So these are individuals that have been moved from a population that is doing well back into a portion of their native range that they've been locally extirpated from. And they found that individuals that are highly exploratory tend to move away from their release site after they're translocated and are much more likely to die. So that's what we're seeing here in this example on swift foxes. The individuals that they measured before they translocated them on exploratory tendency, the ones that spent the most time approached the closest to the novel environment, here down on the bottom, these highly exploratory individuals, were most likely to die after they were translocated compared to the individuals that didn't really go very near to the novel environment at all. They were more likely to survive. So there's maybe a more complex relationship going on with exploratory tendency and novel environments and success in novel environments than we might think. So one possible mechanism for that in my study system, and this is anecdotal, we would have to actually test this, is that it seems like there might be higher predation on our grackle population on the edge of the range. These are two Google satellite maps of our sites. So here in the middle site in Arizona, you can see that it's extremely highly developed. Almost every surface is covered by a human structure, and consequently there are not that many predators on, at least for birds the size of grackles. But in our edge population, it's less developed. Where's my cursor? Okay. So I'll just orient you here. There are some neighborhoods over here, and this is the Golden Corral. This is the shopping center where the grackles hang out. And all of the area that's immediately surrounding the shopping center is open ground covered by natural substrate. So we did see a higher number and a higher diversity of birds of prey at our edge site compared to our middle site. And the grackles react fearfully to these birds of prey. So it's possible that if you're an exploratory grackle in California, you're more likely to get eaten. Some future directions, as I mentioned at the beginning of the talk, I am planning to look at home range size. based on what we've already seen, it seems possible there could be a relationship between home range size and proximity to the range edge, because in Arizona, the grackles are really sticking to specific areas at our study site, whereas in California, all of them are basically overlapping and traveling over a larger expanse. So it's possible this might also relate to exploratory behavior in a way that we haven't looked at yet. I am also collaborating with Dr. Cody Ross, who some of you might know, to incorporate the behavior of grackles around finding a food source into their movement patterns. So this might control for some of the variation we're seeing in movement, where once grackles find food, they might move differently. So this is based on some of the research that Cody has done following around human foragers and looking at their movement behaviors when they encounter a mushroom compared to when they are searching for a productive patch of mushrooms. So some interesting directions that we still plan to go in. Okay, so that is all I have for you guys today. And I am happy to answer any questions that you might have or any thoughts that might have come up for you at this time. Yeah.
You can just pick on anyone who raises their hand.
Yes.
Thank you. Very, very interesting talk. I'm pretty sure I've come across life history-based models of the evolution of animal personality in which there's posited to be a trade-off between thoroughly exploring a small area versus more superficially
Yes.
explored. And but it wasn't one of your predictions that those two things would be negatively related? I mean, I know you found they weren't related at all, but.
So I think that you're talking about fast exploring and slow exploring. So we haven't really looked in depth at those issues, but since we aren't finding any differences in the individual movement behavior within a population, it seems like they're all kind of using, that they're not really showing those two different modes of behavior. I need to look into more about how they like the values, like the cutoffs they use for fast exploring and slow exploring. So this is kind of like a initial overview of these data, and we have a lot more that we're going to look at. We also collected data on boldness. So I didn't show any of you guys that today, but there are a couple different ways to look at animal personality, and so the field is still kind of working out which terms they like the most. So it's possible that the behavior, sorry, I keep touching the mic. The behavior that we're seeing them use to interact with the tent is boldness instead of exploration. So we conducted completely different assays to theoretically measure boldness using like a stuffed Cooper's hawk, which is a known threat. And we plan to compare the performance on these two tasks to make sure that what we're actually measuring is exploration. So there's, in these fast exploring, slow exploring cases, they use a bit of a different method, and trying to compare the behavior we're seeing with the behavior they saw in those tests has been a little difficult. So we're hoping that by making sure that that what we're actually measuring is exploration and not boldness, it can help to piece out some of these things a little bit better.
Thank you.
Yeah, kind of a long-winded answer to your question.
Very helpful.
Yes, in orange.
Fine. Thanks for an interesting talk. I was wondering if you had any, data to supplement sort of the selection of the two different locations with respect to sort of the rate of change of the edge of the population, because that might say something important about sort of like whether there has been, if an edge has not changed or has changed only very slowly, relative to the overall rate of expansion of the range, then that might say something about like selection for or against particular behavioral phenotypes that might not otherwise hold in places where sort of the rate of change of the edge is a lot, quicker, for example.
Yes.
So do you have anything to speak to that?
So what we've talked about so far today is just these areas are places where grackles have either been for a long time or grackles just showed up. There's another aspect of the project that's going to look at the physical environment. So there's an ecological niche model that we're planning to do to kind of map these things about what does it actually look like for grackles in these two places to interact with some place that's less developed and some place that's been developed for a long time. Because they're grackles, so we would see them eating trash in Arizona and we see them eating trash in California. Although there are a lot of natural sites where they hang out in California, they spend most of their days in a parking lot, which is not that different from Arizona. So there's so many questions about that we need to consider further to understand how these species are able to adapt to each new place that they show up. And that's definitely something that we're thinking about.
Cool, thank you.
Thanks, it was a really nice talk. So I'm just wondering about the acclimation to the cage and then putting the
Yeah.
tent in there.
Yeah.
Because it seems like you have a situation where you're putting them into a novel environment
Yes.
and then letting them acclimate to that and then giving them
Yes.
the task of responding. And I'm just wondering, did you record their behavior when you were first putting them into the cage? Because it seems like if you have the food and water, for example, in one corner, right, where they kind of have to be, that you could look at their exploration within
Mhm.
that entire new environment as opposed to a new object to
Right.
explore.
Yes. No, we should do that. We did not.
Could you repeat the question, just for
yeah.
the sake of the folks on?
Yeah, that's a good idea.
Yeah.
So the question was whether we also quantify the behavior of the grackle when they first came into their aviary, in the time when they're habituating to the aviary before we even start behavioral testing. We did not. I think you're totally right. That would have been interesting to look at, and theoretically they should correlate. But yeah, it just didn't happen. We try to give grackles, when they first come into the aviaries, as much like peace and quiet and space as we can because they're freaked out. So we have some data we could probably piece together to look at that, which by that I mean when we start doing their behavioral testing, we measure the amount of time it takes them to habituate to the first test that they do. And so we might be able to get something from that. That being said, as I was kind of indicating, there's not a lot of consistency in how people measure novel environment exploration, and there are pros and cons for each. So a lot of people think that the forced novel environment exploration is not representative of the inherent trait exploration. It might be more like activity, boldness. So we were trying to circumvent that specific thing with this task, but it's not perfect. We also actually did a novel object task, which was similar, except it was just like a pink, fluffy object. They still had food. They weren't food deprived, and they were scared of it. So we stopped doing that. It's clearly not a measure of exploratory tendency. Strange that something smaller was more scary than the tent, but actually the Arizona grackles, there was no difference between their movement in the familiar and the tent condition, showing that they're not scared of the tent, which is important for exploration. So yeah, again, another long-winded answer to your question. Yeah. Yes, Brian.
Do you have any measures of genetic diversity in the two different areas that you're working in? Does that,
Gosh, yeah.
is the idea that you might have less genetic diversity on the edge of an expansion might contribute to less individual variability in exploratory, or less repeatability?
Yes. So we do have genetic data on both.
Sorry, could you repeat it all?
Oh yes,
Yeah.
I'm sorry. I'm bad at remembering.
Yeah.
The question was whether we have any data on the genetics in the two populations and whether we would expect the grackles in the range edge to be more homogeneous maybe in their genetics than the ones in the center. We do have lots of genetic data. I'm not super well informed on that aspect of the project. I'm a not really a molecular biologist, unfortunately, but I know that we are going to look at that. So stay tuned. Yes. Somebody else? Yes.
It seems like you have a trade-off between, well, the good part of exploratory behavior is that you're having more opportunities to find food resources. But on the bad side is that you're more exposed to predators. So is it just birds of prey that are predators, or are there other kinds of predators that typically get grackles too?
Okay, so the question was, oh, I need to plug in my computer. The question was, what are the typical types of predators for grackles? Is it just birds of prey, like I mentioned, or are there other things? So with most birds, feral cats are a huge problem. And similar to both sites, people think that feral cats are struggling and they want to help them. So it's actually really common, much more common than I thought before I started doing this research, for random people to put bowls of cat food in the bushes everywhere. This was so common in Arizona. We would see the bowls of cat food in the middle of a median on a busy road. It's not safe for the cats, you know. So feral cats are a predator for them in both populations, and otherwise it's mostly birds of prey. I think, so just based on the grackle behavior and their, like the life history information that I've read on like birds of the world, birds of North America, snakes can be a problem for eggs and nestlings. And the grackles will mob snakes, but grackles are crazy. Snakes, I have seen several occasions adult grackles approach snakes, even if it's not the nesting season, and attack the snake. So they'll jump at it, they peck it. It could be one grackle, it could be several. So I don't think that they're predators on the adults.
So I was just wondering whether this tent could be viewed as shelter from birds of prey or a
Okay.
danger zone for cats. It sounds like cats and snakes could live in a tent like that.
Yeah, I think so. Would the tent be like a safe stimulus for the grackles, as somewhere they can hide from predators? I had never thought about that, but if there's a predator flying overhead, the grackles will fly under the cars. So I do think that you're right. It could be that because of this escape behavior, the tent might be less scary than we as humans perceive that they should think it is. Yes?
I know the grackles that you were studying were, wild caught and then tagged and everything like that. But do you have any, estimates of the age of the animals that were included in the study as, like a correlate with aspects of movement behavior? Just 'cause I don't know,
Yeah, no, it's a good question. So we measure a lot of things, and so we tried to minimize the additional variables that we had to include by pretty much only catching adults. And in birds, most birds, after they molt out of their feathers from their first year into what become their adult feathers, after that you can't tell how old they are. We did catch a couple of juveniles in both populations. We put them through some of the aviary tests, and they actually performed on the aviary tests. So these are the tests of different cognitive traits, the same as the adults. So by the time these individuals were independent and catchable at our study sites, they at least seem to have the same development of their cognitive abilities as the other adults did. So I'm not sure about whether the juveniles that we followed would differ in their movement behavior to the adults.
Right. I mean, I guess I'm wondering also, like, once they've had that molt, you can't identify, just based on sight alone, the difference between, one that is early in its adulthood versus one that's later on. But is there evidence to suggest that there are sort of like behavioral changes associated with senescence, or is there sort of like just this steep drop off at the end of life?
Yeah, I'm trying to think what's known about grackles. The one thing I can tell you specifically about, the question was, is there anything known about differences in behavior and when behavior of young birds might change into what the behavior of adult birds is? Is that a good?
I guess I'm wondering, like, would aging of the animals play a role in down-regulating the amount of movement that they're having, independent of, how exploratory they are, and whether that's something that's affecting your ability to detect a relationship or not there.
Okay.
But if you were controlling for, if you had an ability to estimate the age of the animals, if you controlled for that, whether you'd see, relationships emerge in a different way.
Yeah, so if we were able to tell specifically this grackle was 3 years old and this grackle was 7, which is what we think is like the average lifespan, what I would expect that we would find based on information from other species, other taxa, is that their movement, the distances that they travel over in this species could go up because they have learned new places to find food. So this is kind of one of the motivating factors behind this additional analysis that we're looking to do with Cody Ross, where these grackles can almost be like traplining hummingbirds, if you guys are familiar with that reference, where they know when certain foods are becoming available in certain places. So I would track them in Arizona, and in the morning they would wake up, leave the roost, and then just in a row hit cat food stations that were on the way to their daily area, their daily home range that they like to hang out. And then towards lunchtime, they're moving towards the cafes. So on the map of the Arizona birds that was on the east side, there was this dense plot of dots just in a straight line. That's because it's the road that borders the east side of campus, and there's a ton of places to eat. So they spend a lot of time hanging out over there. So I think as they age, they start to learn more and more of these things, and it's possible their movement could change in terms of the extent of space that they're using. The flip side is that during the breeding season, all of them kind of contract the amount of space they use to focus around the territory the male is defending, the nest site the female is building, and the young birds, so usually the females, but always the males, don't breed in their first breeding season. So we would expect to see differences in movement behavior based on age during the breeding season, but we exclude the young ones from that one. That's a good question. Anybody else? Yes?
I just had more of a general question, but I was wondering about, I don't know whether grackles are very social, but whether you know anything about whether more social individuals might explore as a group, or I guess the assumption in this is that they're just individuals who are exploratory and those that are not, but I was wondering do they learn socially where to forage, and then groups of them sort of travel together, and could that sort of affect some of the site differences maybe that you found?
Sure, yeah, so that's a good question. She asked whether the social behavior of grackles might affect their willingness to explore new areas and the different places where they hang out. We do include that in the final analysis that we're going to do. So in the pre-registration, we're accounting for social behavior both at an individual level and at a population level. So one of the alternative explanations for a difference in space use on the edge population is that conspecific density is lower. It's an edge, there are not very many grackles there yet, so they are not being excluded by other grackles from any place really. So we're gonna look at that, and then we also do focal follows, which some of you might be familiar with. It's a way to sample behavior and to quantify behavior, where we look at their social interactions, and we're going to create social network maps for each individual so we can look at their social network density and how it relates to their movement behavior. It's a lot more difficult to say at this point in time this individual was with X number of other individuals when it moved to this place. So we don't have that fine scale of data. You could theoretically get that if you had some more like automated tags that recorded the bird's location and any bird that was tagged in that area. So we haven't really gotten that yet. Yeah. Yes, Brian.
One other question about, we had a talk earlier, in fact, about feral cats.
Oh,
in Los
good,
Angeles.
good.
And we had a neat discussion about how the process of trapping, in this case it's to do neutering of feral cats, but that in itself might select for certain kinds or a
Yes.
subpopulation of the total population of feral cats. Is this something that you thought about with regard to your experimental design of how trapping the birds is going to select for certain personality traits perhaps, or, and just kind of as a follow-up, did you find any ordering effects, like birds that were, quote, easily trapped, did they differ in their exploratory behavior in the experiment?
Okay, so the question, so my computer just died. I think this power strip might not be on. So that probably the Zoom
Okay,
feed is.
let's see here, I'm good, so you can speak into this. So, Kelsey is now speaking on a different computer
Sorry.
to answer a question about whether
Well, lots
trapping
of
the birds
technology
influence
going on
selecting
here.
for certain traits.
Yes, that is a huge concern. Sampling bias when you're having to trap wild animals is a huge concern. And so some of the trapping methods that I showed you are like very intuitively going to select for the individuals that are most bold or maybe most exploratory. So you saw there was one in the top picture that was the bow net, where it's this gray half circle and there's food in the middle. Seems pretty low profile, no big deal, but the Grackles were like terrified of it when they first saw it. So we spent a lot of time habituating them to that. There was the large cage also that might select for the most bold individuals. We spent time habituating them to that too. And then there is the mist net, which is probably the only method where there might not be a selection bias based on personality trait. It's a large, it's basically like a large volleyball net that you can spread across a space where the Grackles typically fly. And the material of the net is very fine, so they can't see it. In fact, people can't see it. In Arizona, we several times had people walk into the net. Somebody mowed through it with a lawnmower. We had somebody ride their bicycle through it. So we got this question on one of our pre-registrations, and we compared the performance of the Grackles and the aviaries that were caught with the bow net and those that were caught with the mist net, and there was actually no difference in exploratory performance or in performance on some of our other tasks based on trapping method. So we have a consistent sample. Hopefully that means that we didn't have a selection bias because we spent so much time habituating the Grackles to the bow net and the walk-in trap. This particular question, with the way that we're comparing two different populations using the same trapping methods, hopefully we can still count on our answers or our results between the populations because it would be the same potentially biased sample.